What is a cell? Unicellular organisms are capable of first, independent existence, and second, performing the essential functions of life. Anything less than a complete structure of cell does not ensure independent living. Hence, cell is the fundamental, structural, and functional unit of all organisms or living organisms. Anthony van Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of microscope and its improvement leading to electron microscope revealed all structural details of the cell. Cell theory. In 1838, Matthias Sheldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which forms the tissues of the plant. At about same time, Theodore Schwann, a British zoologist, in 1839 studied different types of animal cells and reported that cell had a thin outer layer, which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Sheldon and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow in 1855 first explained the cells divided and new cells are formed from pre-existing cells, commonly termed as omnicellula e cellula. He modified the hypothesis of Sheldon and Schwann to give the cell theory a final shape. Cell theory, as understood today, is first point, all living organisms are composed of cells and products of cells. Second, all cells comprises from pre-existing cells. Overview of cell. You have earlier observed cells in an onion peel or a human cheek cell under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer boundary as the limiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosomes, which in turn contain the genetic material called DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are called prokaryotic. If both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical structures and reactions occur in it to keep the cell in the living state. Besides the nucleus, The eukaryotic cells have other membrane-bound distinct structures called organelles like endoplasmic reticulum (ER), the Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria, and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. Cells differ greatly in shape, size, and activities. For example, mycoplasmas, the smallest cells, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cells. The prokaryotic cells are represented by bacteria, BGA, blue-green algae, mycoplasma, and PPLO, pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus or spherical, third, vibro or coma-shaped, and fourth, spirillum or spiral shape. The organisms of the prokaryotic cell is fundamentally similar even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is called the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to genomic DNA, the single-cell chromosome or circular DNA, many bacteria have small circular DNA outside the genomic DNA, which are commonly known as plasmids, which are used in biotechnology. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher class, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions, which are a specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. These are essentially enfoldings of plasma membrane, cell envelope, and its modifications. Most prokaryote cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is, the outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of the envelope performs distinct function, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in the cell envelopes and the manner in which they respond to staining procedure developed by Gram. Those that take up the Gram stain are called Gram positive, and the others that do not are called Gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. The membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extension of plasma membrane into the cell.